This is a supplementary lecture that has been recorded for advances in molecular biology. It's discussing the applications of proteomics in systems biology. Now, it is part of a... Why aren't you advancing? There we go. Now we're advancing. Okay, so back at the ranch. This is extending on from some other videos that I've done. If you've been on the YouTube channel or if you're doing analytical biochemistry, you would have seen these before. These other videos are far more technical. They're discussing the technical and practical aspects of doing proteomics. So there's this introduction to proteomics, which talks about how to do sample prep and the kind of separation technologies that you can actually use. And then there's another one where there are 6 videos discussing mass spectrometry, and a couple of those are specifically about using mass spectrometry to do proteomics. This video is a little bit different. We're going to concentrate on some different topics and mainly about what you can actually do with proteomics. So if you're interested in the technical aspects, go back and have a look at these. So I'm just going to remind some people about what proteins actually are, and don't worry, I am actually going somewhere with this. Proteins is a generic term. Okay, it can mean a lot of different things depending on the context of when you say it. But at its simplest, it is a term describing a polymer or a string of amino acids that then form a 3D structure that then allows a function. So we have our 20 different amino acids here. Some of them are non-polar or they're hydrophobic. Some of them are polar and can form hydrogen bonding and disulfide links with other amino acids within the chain. And then some of them actually form ionic in solution. So you've got these negatively charged acidic amino acids and these 3 basic amino acids that can all be either positive or neutral, or in the case of the acidic ones, negative or neutral, depending on the pH. These are then put together as a polymer where you get the end, what's called the amino terminal or the N terminal, where you've got this primary amine, and then the C terminal or the carboxylic acid group is then linked in an amide bond to the next amino acid. So the amino acids are defined by their side chain within the sequence. So here we have tyrosine, here we have threonine, we have glutamate, tryptophan, isoleucine, and then the C terminal of the peptide. These things can be very, very long. So typically there is a blur at where the difference between a peptide and a protein actually occurs. So anywhere up to about 100 amino acids could be a peptide, but then getting larger and larger, they become a protein. So these amino acid strings then form secondary structures by using hydrogen bonding or hydrophobic interactions, or they can form ionic bridges, or they can form disulfides to then assemble themselves into alpha helices and beta sheets, then into tertiary structures, sometimes with metal cations bound inside them or other molecules, and then they can assemble into protein complexes. In short, the amino acid sequence determines the structure of the protein, whether or not they form helices or sheets, the way that some amino acids from one side of the protein chain can bind to the other side of the protein chain. But this structure and folding then determines the function of the protein. Now, some of this can be predicted by looking at the gene sequence, but not all of it. So now I want to move on to this term describing proteoforms, because the term protein is actually a really poorly descriptive term for stuff that's actually really, really complicated. And so a proteoform is a much better term. And what it means is that it is all of the different molecular forms that can come from a single gene. And I'll talk about this more on the next slide. And so it allows you to include genetic variations, alternative splicing, and most importantly, post-translational modification, which can include proteolytic cleavage. So every time that you get a modification to a protein chain, you form a new proteoform. So we describe this a little bit better in this slide here, where in the databases such as UniProt, there are these what are called canonical sequences, which is the open reading frame inferred by the bioinformatic translation of the genome. So bioinformatics software goes through, it finds methionine start codons, and then it works its way through until it finds a stop codon, and then it translate that into a protein sequence. But there's actually no evidence at this point whether or not there is a protein or a proteoform in existence. It is just a translation. Now, we can get this sequence being produced by the ribosome, and the primary sequence that's produced by the ribosome is actually a proteoform. Now, we can have things like RNA splicing, we can have short nucleotide polymorphisms, we can have mutations in the DNA, and all of those, when translated, will create a different proteoform. So if we have a gene that gets translated to RNA, but that RNA gets a piece of it spliced out, that is able to then form a different proteoform. You can also get proteoforms produced by proteolysis. So the primary sequence gets translated by the ribosome. When it gets exported, you get things like signal peptides cut off and other cleavage sites. And every time that happens, it creates a new proteoform. So then you can have post-translational modifications where we add phosphorylation or we add methylation, and each time that this happens, we form a new proteoform. None of this is predicted by the genome. So this is yet another example of what I was just talking about, where we have transcription, we have isoforms and splice variants. These things can form distinct isoforms. They then can undergo different modifications and create different proteoforms from a single gene sequence. This stuff is not predicted by the genome. One more time, we have our primary amino acid sequence from the ribosome. If that is left unchanged, that is one type of proteoform. If we do something like ubiquitinate it, we have now a different proteoform from the original one. If we phosphorylate, and if we have one phosphorylation, it's a different proteoform from the original. If we have two phosphorylations, then it is not only a different proteoform from the original, but a different proteoform to the other phosphoform. If we have one of these where we have a methylation and a phosphorylation at a different site, we now have yet a different proteoform. Now, the reason that I'm talking about this, and I'll come back to this again on the next slide, is this part here where to do shotgun proteomics, what we are doing is we are taking proteins and we are turning them into tryptic peptides. And as you can see, some of these peptides are actually shared by different proteoforms. And so now, if we only detect the proteoforms that are not defining the proteoform, if we're only detecting the common peptides, we can't determine that there were multiple different proteoforms. So this is what I'm talking about in a summary. The majority of proteomics experiments are actually done by peptide-centric methodologies. This is measuring the abundance changes in open reading frames, not in mature proteoforms. And so the abundance of these open reading frames is inferred by the abundance of peptides that come from the cleavage of proteoforms that are in the sample. But once you then go through and you analyze this, you may not get any proteoform information. Because if you don't measure and identify the peptide that is defining that proteoform, you don't know it's there. Like I said, there's more information about how this works and how you can do this on the other videos on the YouTube channel. So now that I've discussed that, we want to actually discuss just quickly what proteomics can actually do. And so what proteomics' main aim is to measure the change in abundance of proteoforms, ideally of proteoforms. Unfortunately, the majority of proteomics projects measure the changes in abundance of open reading frames. That information is still valuable, but ideally we would like to focus on proteoforms. So we want to do this to evaluate the effect of things like the mutations of genes in cancer, changes in environmental conditions such as temperature, pH, different concentrations of metabolites and other molecules, the effect of growing things in different nutrients. We also want to use this in biomarker discovery to look for things, to look for markers of disease that we can use either as a diagnostic or as a lead to develop drug treatments for various different diseases. Most importantly, proteomics is unbiased and untargeted. Because a lot of other methodologies that are used to measure proteins are not necessarily measuring proteoforms, or they are only targeting a very small number of these. So antibody-based assays such as Western blots and such as ELISA and immunofluorescence microscopy are only looking at specific proteins, not at all of the proteins in an unbiased manner. Now, I've taken this little quote here down from this article that's listed down at the bottom here. My mouse stopped moving for a minute, I apologize. Where it actually talks about how we can put this into clinical applications, because nowadays we can measure the proteomes from formalin-fixed paraffin-embedded tissues, which is what happens to a lot of tumors that are isolated from people during surgery. We can look at the proteome of blood, of plasma and serum. We can look at different body fluids to measure the concentration of different proteins. We're starting to be able to do this in single cells, to be able to figure out what happens during crosstalk between different cells of a particular organism or a particular tissue. So I just want to run through two examples that can show you how proteomics can actually be applied. And the first one is this examination of what happens in plasma of patients that have been infected with COVID-19. So what are the proteome-wide effects of being infected with COVID? So this was done in Munich. It's a relatively small study, but it's a relatively large study when you think about proteomics, where they have 31 patients and they're following them over time. This is actually very, very important for this study, and they're profiling their plasma proteome, and this is actually really difficult, and we'll come back to this in the next slide. So they had, of those 31 patients, they took numerous blood samples from people who were PCR negative for COVID, but they had all of the symptoms that you would normally associate with COVID. It's just that they didn't have COVID. There were 31 people who actually did have COVID, and they followed them over the course of 54 days. So they took a lot of samples. So there were 262 samples from these PCR negative people, and there were 458 samples from these COVID-19 infected people. By analysing their plasma, by taking a microliter, isolating the proteins, turning them into peptides, putting them into liquid chromatography, tandem mass spectrometry, and doing data analysis to look at what the fold change and the significance of those fold changes was, they measured across all of the cohorts 502 proteins and discovered what they considered to be 71 different biomarkers. And as I mentioned before, doing plasma proteomics is really actually very, very difficult. Yes, they found 502 proteins reproducibly, but there was only about 300 in each of those patients. And this reflects the problem with plasma's dynamic range, which is illustrated in this diagram here, where you've got 10 orders of magnitude of concentration. And so the first 400 or 500 proteins are all up in this region here, the really high abundance proteins. You're not measuring cytokines or interleukins. You're not measuring things from down here. You're measuring the changes in abundance of the most abundant proteins in the serum. But surprisingly, this is still very, very powerful. What they found was that over the time course of following these patients, there was a downregulation in the innate immune system. There was an upregulation in lipid homeostasis, and more importantly, there was an upregulation in proteins related to coagulation. And overactive coagulation is a main complication in people who have COVID. Really interestingly was this finding here, where this particular protein here, which is known as a pro-inflammatory protein, is significantly increased on the first day that they are sampled. And if it is significantly increased on the first day of sampling, they are more likely that they are not going to survive COVID. So this is now a possible predictive classifier for mortality. So if we started to take plasma and measure for this protein on the first day that someone presents in hospital, we may be able to help them to survive. And this protein has been independently confirmed as a predictor of mortality, and this validates this proteomic analysis. One of the other things that was observed is how the body undergoes seroconversion from the initial IgM response to IgG responses. And the other interesting thing is that the proteomics is able to detect immunoglobulin regions in patients that are otherwise showing negative immunoassay results. So they were examining 5 different immunoassays. Sometimes they had patients that had a negative result but were PCR positive, and they were able to detect immunoglobulin regions related to COVID antibodies within their samples where they couldn't find them by the immunoassay. So that's the first example of what you can do with proteomics in a disease state. The second example is another serum proteomics project where they're looking at the effects of high-intensity exercise. And there's a number of these shotgun proteomics approaches, but I wanted to pick this one because it shows the other main way of doing proteomics, that of doing 2D gel electrophoresis, where the proteins are first separated out by isoelectric point, the pH at which they are neutral, and then they're separated out by size. So each one of these spots is a single or a collection of proteoforms that all have the same isoelectric point and the same mass. If a protein or a proteoform has the same composition but the amino acids arranged in a different way, they will have the same isoelectric point and the same mass. So they will focus to the same place. But interestingly, because we're separating on the proteoform level, what we are doing is things that are post-translationally modified, that are cleaved or acetylated or phosphorylated, the position of the spot will move. So this is one gel of an example. They had 6 different patients. They measured, they had a gel for pre-exercise. They had an individual gel for each person for 5 minutes post-intensity exercise, 1 hour post-intensity exercise, and then they found that there were 20 spots that were differentially abundant. Some of them had gone up 1 hour post-exercise, some of them had gone up 5 minutes, some of them had gone down. And you can see the change in proteome abundance by the color that is indicated in the indicator down here. What you find from this study when you look at these proteins is that you can group them together and they start to show interesting things with response to exercise that could explain some other observations that have been made in other studies, such as there is a modulation of proteins that are involved in immune regulation, and these may show that high intensity exercise has a role in prevention and treatment of many chronic diseases that are related to immune system modulation. Anti-inflammatory effects are mediated by actual different proteoforms of fettuin and antitrypsin. So these are the same gene sequence, but they have been modified into different proteoforms, and those proteoforms are specifically increased or decreased when high intensity exercise occurs. The increase in apolipoproteins that was seen is related to the handling and clearance of cholesterol, looking at the modulation of high-density lipoprotein and low-density lipoprotein, and this could be a means of preventing systematic inflammation. So proteomics can reveal this, and you can relate it to this. Also, the increase in 3 clusterin proteoforms following acute exercise may show why lower levels of heart disease are seen in individuals that are more active. And again, it is proteoform specific, not gene specific. So you can see that in these particular circles here, the changes in biological function that happens in the plasma samples, where you've got differences in amyloid beta binding, you've got increases in HDL, LDL particles, antioxidants, heat shock proteins, and vesicle formation. And then in some of these associated pathways that involve these particular proteins that are changing in abundance, signaling, mineral absorption, and again, high density lipoprotein remodeling. So that's just a brief introduction as to what we can do with proteomics, but it's only one measure of the phenotype of different cells and different organisms. And other measures of the phenotype are things like metabolomics, how does the amount and concentration of metabolites change, lipidomics, fatty acids and phospholipids and such. How do molecular interactions change with phenotype? How do protein-protein interactions change? How does the assembly of protein complexes change? What happens when to change protein and DNA, protein and RNA interaction? What happens when metabolite levels increase, and how do they change protein abundance? And we want to combine all of these measures of phenotype in systems biology to have this holistic approach to personalized medicine, where we know that one person treated with a certain amount or type of drug is actually going to have a beneficial effect, but with another person, it may have no effect at all, or it may be detrimental. So we want to use systems biology to try and overcome those problems and tailor particular treatments to particular people by examining their blood, their urine, their saliva, proteome, metabolome, lipidome. It also has application in biotechnology with regard to things like plant breeding, antimicrobial resistance. There's anything that has proteins in it, you can apply these methodologies to and find out biological information.